2.8.5 Sperm competition. Once females have mated, there is always the chance that a subsequent male will mate with her and displace the sperm in her spermatheca from a previous mating. Sperm competition results. This chance is maximized when 1. Females mate several times before eggs are fertilized. 2. Inseminated sperm is stored by the female in the spermatheca. 3. Sperm remains viable for a long time. 4. Sperm is used economically, without any waste, and often one sperm is used to fertilize one egg. Parker 1970a. Under these conditions, strong intersexual selection will result, with inevitable adaptations in males to minimize sperm competition. These strategies include the following and have been reviewed by Parker 1970a and Thornhill and Alcock 1983. 1. Mating plugs block the genital passages of females after copulation, preventing or reducing possibilities of subsequent copulations. They are formed by secretions of the male accessory glands and are common in Diptera, Hymenoptera and Lepidoptera, but have been observed also in Orthoptera, Coleoptera and Isoptera. The plug may dissolve in a few hours, but probably lasts until a female becomes unreceptive, or it may even induce unreceptivity. 2. Prolonged copulation is common in insects. Insemination may be rapid, but copulations last well beyond this necessary process, to 30 minutes, 60 minutes and even 4 hours, just within the Diptera. Thornhill and Alcock 1983. In the notorious lovebugs, Plachini arctica Diptera, Bibionidae pairs may remain in copula for up to 3 days. Moths commonly mate for 24 hours. Clearly, males prevent further copulations while joined, and they may induce unreceptivity in females, but forfeiting additional copulations appears to be a high price to pay. Perhaps, when competition for females is high, assuring one successful copulation is a better strategy than going for another female, and risking sperm competition in the original mate. 3. Tandem positions or passive phases occur when males remain attached to a mated female but without genital contact. Males defend females against other males, often while the female oviposits. Therefore, post-copulatory passive phases reduce sperm competition. In the dung fly, Scatophagus stercorea, males mate and then guard females on fresh dung, sitting on the back of the female. Without genital contact, the female can oviposit into the dung, but the male assures that his sperm fertilizes her eggs. Should he be displaced, a second male will fertilize 80% of the eggs she lays. Parker 1970a, B, C. Tandem positions are adopted in locusts, grasshoppers, most crickets, and in many dragonflies and damselflies. 4. Non-contact guarding phases involve males which guard females they have copulated with, but the pair do not remain in contact. A female may remain in the territory of a male which defends against entry by other males. Both sexes may then be opportunistic in mating, and if populations are dense, such opportunities are rich, with extraterritorial males sneaking copulations with females while the guarding male attends to other females which have entered his territory. In the damselfly, Colopteryx maculata, males can actively remove sperm in the spermatheca of a female with a specialized organ analogous to a penis, and replace sperm with his own. Wage 1979. Non-contact guarding has been noted in dung beetles in this chapter, and it also occurs in bark beetles and crickets. 5. Takeover avoidance includes any mechanism that improves a male's probability of inseminating a female and ensuring that his sperm will fertilize her eggs. Tactics may include clasping appendages in the male genitalia which improve the strength of attachment to a female. Prothoracic legs of males may be modified to grasp females firmly, as in the Didicidae, the predaceous diving beetles, with a segment of the tarsus expanded to house suction discs for firm attachment to females. Rejection behaviors by mated males may include kicking at intruding males as in grasshoppers and scatophagid flies. Emigration from dense assemblages of males reduces intramale competition. In the suborder of Diptera, the Nematocera, the longhorned flies, such as mosquitoes, black flies and midges, pairs form in a swarm, but may drop to the ground to copulate. Male dung flies, Scatophagus stercorea, carry females from dung to surrounding grass with increasing frequency as male densities on dung increase. Figure 2.18. In the cooler conditions away from dung, duration in copula is prolonged, but reduced probability of takeover and reduced disturbance provides a net benefit to fitness. Parker 1971, Borgia 1980, 1982. Darwin 1871 observed a large range of secondary sexual characters of arthropods from crustaceans to spiders and insects, spending almost 100 pages on this group with most emphasis on insects. He concentrated on visible characters that differed between males and females, which were under the influence of sexual selection. What he did not appreciate was the vast range of behavioral adaptations involved, and the many more cryptic tactics employed in such activities as sperm competition, alternative mating strategies and nuptial gifts. The subject of sexual selection has enjoyed a resurgence since the Centennial Symposium on the subject, Campbell 1972, before which little progress had been made since Darwin's time. Now, as covered briefly in this chapter, we can see how much studies on the insects have contributed to advances in knowledge on this fascinating subject. Many of the intricacies of sexual selection have been revealed, and yet, no doubt, there are rich research opportunities for the future. 2.8.6 Parental investment and care of progeny. Trivers 1972, page 139, defined parental investment as any investment by the parent in an individual offspring that increases the offspring's chance of surviving, and hence reproductive success, at the 15. 0. 30 60 20 25. Figure 2.18 Alternative mating strategies in the dung fly, Scatophagus stercorea, which uses fresh, warm dung as mating and oviposition sites. A. As density of males increases on dung, more males carry females away from the dung to mate in the surrounding grass. The arrow indicates a possible threshold density for emigration. B. The duration of copulation among emigrants is prolonged relative to those on dung because of the reduced temperature in the grass. From Parker 1971, A. Modified B. Reprinted with permission from Blackwell Publishing Inc. Cost of the parent's ability to invest in other offspring. Parental investment therefore includes the metabolic cost of primary sex cells as well as provision of food for young and protection of young. In most insects, males provide nothing for progeny except sperm. But some males provide nuptial gifts which contribute to the welfare of young, and some males help to feed young and/or guard females against takeovers as we have seen. Among female insects, the majority simply lay eggs without further guarding or other investment. However, many females are meticulous in placing eggs in suitable places, spending considerable time finding oviposition sites advantageous to their young. In doing so, they become exposed to increased predation risk, with the reduced potential for further reproduction. Many groups, in addition to a complex chorion, provide protective coverings for eggs, such as oothecae in roaches and foamy egg cases or oothecae in mantids, or females cover clutches of eggs with protective secretions, for example in some membracids and reduvids. Some stick insects produce a capitulum on the operculum of the egg which is attractive to ants, like the elaiosome on seeds, stimulating ants to carry the eggs into the safety of their nest. The function of nuptial gifts from males to females is difficult to evaluate without careful tracking of nutrient use by females. In many cases, the nuptial gift functions solely as a mating investment rather than a contribution to progeny welfare. However, detailed studies on some species show evidence of considerable male parental investment. Thornhill and Alcock 1983, Thornhill and Gwynn 1986. Spermatophore constituents provided by males to female butterflies are used in egg production, helping to maximize egg number. Boggs and Watt 1981, and advancing early egg maturation when pollen sources are sparse in Heliconius and Danaus butterflies. Figure 2.19, Boggs and Gilbert 1979. A Heliconius butterfly male, with one spermatophore, can provide enough nitrogen to supply 15 to 30 eggs. Grasshopper males also pass spermatophores to females with nutrients rapidly absorbed into developing eggs, and multiple matings increase fecundity in Melanoplus sanguineps. The 5-hour-long copulation seems to be mostly adaptive for transfer of nutrients, given that sperm can be transferred in 10 minutes. Male insects may also show parental care, but rarely. In Nigrophorus carrion beetles, males will help bury the corpse of a bird or small mammal, excavate a burrow beneath the corpse, masticate corpse material to form a nest in which the larvae develop, and in some species males will aid females in feeding young. Wilson 1971. In some bark beetles, males and females collaborate in excavating tunnels and caring for the brood. These 1, 2, 3, 4. Number of previous mates of male. 0%. A 10. Figure 2.19. When males of the butterfly, Heliconius sydno, have mated little before mating with a female, they provide a large spermatophore, with the result that females wait longer to mate again, and lay more eggs fertilized by the male's sperm, compared to waiting times after mating with males which have mated with many females. Data by C. L. Boggs. From Thornhill and Alcock 1983. Reprinted by permission of the publisher from The Evolution of INSECTMATING Systems by Randy Thornhill and John Alcock, p. 384, Cambridge, Massachusetts, Harvard University Press, copyright #1983 by the President and Fellows of Harvard College. Kinds of behaviors qualify as subsocial. Adults care for their own progeny for a period of time. Wilson 1971. In Chapter 10 on Life Histories, another case of male parental care is discussed, the giant water bugs, where males provide the care exclusively. Females are more likely to provide parental care because they invest far more than males in their progeny, and therefore they are more limited in the number of progeny they can produce. See Wilson 1971. Female care occurs in the Hemiptera, where females guard egg clutches and young. Crickets may dig a burrow in which they feed their young, and many beetle families have species with subsocial behavior. Perhaps the greatest contribution females typically make toward progeny welfare is the careful placement of eggs. Females are frequently particular and specialized in placing eggs on or in suitable host plants or animals, as illustrated in figures 2.3, 2.4 and 2.5 and in Chapter 4 on plant-insect interactions. Some kinds of insects can even decide on whether to place a male or a female egg in a particular position, depending on the resource quality available. In the Hymenoptera, for example, reproduction is haplodiploid. Males develop from unfertilized eggs, they are haploid. Fertilized eggs become diploid females. This condition has arisen several times in primitively herbivorous insects, including the Hymenoptera, with sawflies at the base of the phylogeny. It constitutes a form of parthenogenesis known as erinotoky. Males have no fathers. 4 insect orders, with many herbivorous species, include examples of haplodiploidy, Hymenoptera, Hemiptera, Homoptera, Thysanoptera and Coleoptera. Several families of mites are also haplodiploid, including herbivorous spider mites, Tetranychidae, White 1973. The condition has arisen several times in herbivorous groups, but its adaptive value has remained mysterious. However, 2 studies have shown that sawflies allocate sex ratios in an adaptive manner. Craig and Mopper 1993. Tenthretinid sawfly females lay a greater proportion of female eggs on rapidly growing plants, in which survival is better. Females are larger than males and larger females were more fecund than on slower growing plants. Hence appropriate sex allocation improved female fitness. Craig et al. 1992. The second study concerned a diprionid sawfly which laid a higher ratio of female eggs in trees supplemented with water and fertilizer than on trees that were not treated. Mopper and Witham 1992. On the treated trees, females attained a greater mass, and being pro-ovigenic, having all eggs ready to lay on emergence, they were more fecund. Much more work has revealed advantages to sex allocation in haplodiploid parasitoids where female wasps are originarily larger than males, and female eggs are more frequently oviposited into larger hosts than male eggs. Figure 2.20, Clausen 1939, Charnov 1982. What we learn from the study of insect behavior is that detailed observational studies combined with carefully executed experiments are needed to unravel the intricacies of insect life histories. See also Chapter 10. Given their existence on Earth for about 400 million years, since the early Devonian epoch, Grimaldi and Engel 2005, perhaps we should not be amazed at the complexity of interactions between the sexes of insects and their resources such as plants and animals. However, the literature reveals repeatedly fascinating and unexpected details on the life of insects, with many more surprises no doubt awaiting discovery. No wonder then that Vladimir Nabokov, 1966, page 126 wrote, "Few things indeed have I known in the way of emotion or appetite, ambition or achievement, that could surpass in richness and strength the excitement of entomological exploration." Thomas Eisner, 2003, page 1, admitted a lifelong thrill of discovery. In me, love of nature is expressed as an affection for insects. I am an incorrigible entomophile. Edward Wilson, 1994, page 191, wrote, "Love the organisms for themselves first." Quote. He concentrated his attention on ants in particular, but these authors illustrate the dedication and focus that made them exceptional. Behavioral studies of insects were of central concern. 1.0, 0.8, 0.6, 0.4, 0.2, 0.6, 0.8, 1.0, 1.2. Applications. Behavioral approaches to pest regulation. With increasing problems concerning insecticide use, insecticide resistance, environmental contamination, biological concentration, population rebound, non-target species, the discovery of effective alternatives for insect pest population regulation became imperative. Therefore, any methods, or their combination, that impair activity of pest insects, or improve the effect of their natural enemies, using good biological knowledge of field populations, have become a major focus of research and application. Semiochemicals of all kinds hold promise in behavioral control of insect pests, e.g. Nordland et al., 1981. Fortunately, semiochemicals are generally highly pest-specific and should cause little or no adverse effect on non-target organisms. Thus the prospects of managing many of our major pests by taking full advantage of semiochemicals shall be welcomed by environmentalists and the public who are increasingly concerned about the maintenance of environmental quality. Knipling 1981, page she. Added to semiochemical use are visual attractants for visually hunting pest species, such as apple maggot flies. Provision of food for natural enemies in or around crop plants is also potentially an important contribution to pest control. The use of plants that provide nectar and pollen, and extrafloral nectaries, can be added as indirect defense of a plant crop to the range of regulatory methods for suppression of insect pests, e.g. Wa diaresis Kurz et al., 2005. Many other methods may be integrated into a general strategy of pest regulation involving such approaches as landscape management, hygienic maintenance of farmland and forest, maintenance and enrichment of plant biodiversity, and application of knowledge of multiple trophic level interactions, e.g. Charnke and Dawkins 2002, Charnke et al., 2007a. When an insect pest is well understood, physiologically, behaviorally and ecologically, methods may be combined into a comprehensive approach to pest regulation, now known as integrated pest management, IPM, e.g. Pedigo 2002. This involves integrating methodologies for all the major pests on a crop throughout the season, with the key to success being the understanding of their biology. The integration of cultural, behavioral and biological controls is well illustrated by research on commercial apple orchards by Prokopi and his research group Prokopi et al. 1994. Blending decades of research on apple pests, see Prokopi 1997, Prokopi et al. 1994. Prokopi and coworkers devised a six-pronged IPM approach involving management of all classes of pests in apple orchards in Massachusetts, USA. 1. Monitoring of pest populations using visual traps or direct observation was essential in determining phenology of pests and densities, to decide if early pesticide treatment was necessary, and when to apply it if necessary. Pesticide use was minimized and restricted to early season use, which permitted natural enemies of pests to colonize and multiply through the late spring and summer. 2. Apple maggot, Ragoletus pomonella, Diptera. Tephrididae was regulated with the use of a behavioral method. 8 cm red spherical sticky traps baited with a synthetic fruit attractant and a synthetic food attractant placed every 5 meters on perimeter apple trees. The spheres provided supernormal stimuli for the fruit flies, and immobilized the flies on the sticky surface. 3. Hygiene was used to control codling moth, Sedia pomonella, populations by cutting down all apple and pear trees outside the orchard for 100 meters around, a distance rarely traveled by adult moths. 4. Hygiene was also employed to prevent buildup of codling moth and other pests after apple harvest, by removing dropped fruit. 5. No direct control methods were used against such pests as leaf rollers and scales, because natural enemies became numerous enough after the early insecticide spray. 6. A blend of herbaceous plants was grown among trees which favored populations of spider mites, Tetranicus urticae, and the predaceous mite, Amblysius folicis, providing a reservoir of predaceous mites from which apple trees would be colonized. The development of this integrated approach was based on a large research program, much of it behavioral in nature, and investigations continue, e.g. Rull and Prokopy 2005. The methods are relatively labor-intensive, but the benefits of using more natural approaches to pest regulation are less toxicity in the environment, little impact on non-target species, reduced possibilities of evolved resistance to the methods applied, higher biodiversity and an agricultural ecosystem regulated in a more sustainable way. Another successful program, reported by Zhang et al. 2008 targeted one species, the melon fly, Bactrocera cucurbitae diptera, Tephrididae in Hawaii. Malatrapping, bait spraying, sanitation of crops, the sterile insect technique, cibolo, and augmentation of a parasitoid were all employed in this effective integrated pest management plan. Many other applications of behavioral methods of pest control have been employed, e.g. Judson and Gordon 1989, Ridgway et al. 1990, Pedigo 2002. Pheromone traps may be used for monitoring insect populations to determine the need for regulatory methods and their timing. Pheromones and plant derivatives may also be used for mass trapping. Coupled with insecticide in the bait, large numbers of insects can be killed, with successful control of some fruit fly species, cotton boll weevil, Anthonymus grandis, bark beetle species and ambrosia beetles, Lanier 1990. Mating disruption can be effective by swamping the environment with pheromone from many point sources so that these compete with females releasing pheromone and males cannot discover mates. Pink bollworm, Pectinophora gossypiella, was the first species to be controlled successfully at the commercial level using mating disruption, CARD, E1990, and codling moth has been effectively controlled, Whitskall et al. 2008. Other species include oriental fruit moth, Graphilitha molesta, and gypsumith, Lymantria dispar. Behavioral modification methods have been used in horticulture, forestry and in field crops, and even in the regulation of medically important pests as CC flies, Glossina species, Ridgway et al. 1990. Other methods of pest regulation also employ a detailed understanding of insect behavior. For example, the regulation of the New World screwworm fly, Cochleomia hominivorax califoridae, which was a devastating pest of cattle in the southern USA and Mexico, depended on male and female behavioral characteristics. Females mate only once, meaning that mass releases of sterile males, induced by irradiation, can inundate a population, resulting in most females becoming infertile. The sterile male technique, e.g. Pedigo 2002. Males may mate several times so that sterility is propagated effectively. When the method began to fail, it became evident that mass rearing had reduced male competitive ability and general competence, so more effort was devoted to frequent injection of wild-type males into the mass-reared populations. Continual monitoring of male behavior is essential when mass rearing is practiced. Sterile insect mass rearing is now a global enterprise, including many fruit fly species, the screwworm, moths, such as codling moth and pink bollworm, and CC flies, Hendricks and Robinson 2003. Many behavioral studies have involved the understanding of foraging by herbivores, predators and parasitoids in a quest to evaluate searching efficiencies, e.g. Wahage and Greathead 1986, Hassel 2000. These are frequently in the interest of biological control programs with some work already discussed in this chapter in the section on foraging behavior. Studies were undertaken on glasshouse pests where predictions on activity and effects of interactions could be predicted well in such controlled and enclosed environments, e.g. Dick 1988, Grostel and Dick 1999, Cripps et al. 2001. Conservation biology necessarily involves behavioral ecology, see Samways 1994, 2005. The behavior of insects informs us of their most profitable habitats, where they go in winter, and the quality of resources most beneficial to a population's survival. Sensitive resources, habitats and landscapes may be identified. Without knowing the migration routes of monarchs, it would be impossible to protect the butterflies where logging threatens winter roosting sites in Mexico. Once these sites were belatedly discovered, a monarch butterfly biosphere reserve was established, which